Okay, so regardless of the weather, it's 8:01 and I'm going to go ahead and get started. So I want to just say today, I hope you had a safe drive in. And my fact for today is a really positive weather fact for once, and that is that the sun does not set tonight until 4:59. So we're going to break the 5 p.m. setting over the weekend. The days are getting longer, and spring is on the way. So with that, I want to tell you about our grand rounds today. Thank you again for taking an hour of your time to learn more about science, more about medicine, and just make yourself better. Our grand rounds today is brain cognitive Alzheimer findings. And we have Dr. Sterling Johnson, PhD, and Dr. Tobey Betthauser, PhD, MS, giving grand rounds today. And before I introduce them, I want to let you know that they stepped in with a week and a half notice to do grand rounds. I just want to thank them. So, for all those in the audience who wonder how grand rounds happen so seamlessly, behind the scenes, oftentimes a little glitch will happen here and there. And I will say I'm so thankful that there are people who are willing to step up, even with their arm twisted just a teeny bit probably, to just come and talk about the work that they do. And certainly you know that we are one of the epicenters of research in Alzheimer's disease and one of the best in the world. So with that, I'm going to introduce our two speakers today. Our first speaker is Dr. Sterling Johnson. Dr. Johnson received his Doctor of Philosophy at Brigham Young University. He then went on and did a pre-doctoral internship with a neuropsychological emphasis at Dartmouth Medical School in the Department of Psychiatry, and then did a postdoc fellowship in clinical neuropsychology and functional neuroimaging, also at Dartmouth Hitchcock Medical Center. He spent the first part of his career in Arizona, and we were very lucky to have him join us here at University of Wisconsin in 2002. And he holds several leadership positions for us here. He is the Associate Director for the Alzheimer's Disease Research Center. He is also the Associate Director for the Wisconsin Alzheimer's Institute. He holds the Jean R. Finley Professor of Geriatrics and Dementia Professorship, which is a great honor. He is incredibly well published. When I was reading through his CV, and I just wanted to confirm with him this morning, he participates in 6 R01s, and of which 4 of those he is the PI or the multi-PI, and he is the site PI for one other, and then he's the Associate Director for a center grant. So that's really incredibly remarkable. He's had several great honors in his career, and they revolve around research, which obviously is his areas of expertise. He received our Rankin Research Award for significant research accomplishment by a senior faculty member in the Department of Medicine. That's really one of our most coveted awards. And he's also received Tomorrow's Leaders in Alzheimer's Disease Research Award from the Alzheimer's Association. Our second speaker today, who I didn't meet until this morning, was very lucky to meet, is Dr. Tobey Betthauser, PhD, MS, and he did his Doctor of Philosophy and Master of Science in Medical Physics here at University of Wisconsin. He then has gone on and done his postdoctoral fellowship in Alzheimer's disease biomarkers, cognition, and risk factors, which you'll hear some about today, at our ADRC, which is the Alzheimer's Disease Research Center. We were lucky enough to have him join us here as an associate scientist, where he works closely with Dr. Johnson. He also is participating and active in 4 grants, including 3 R01s, which are the R01s that Dr. Johnson has. And when I thought about looking through something really interesting on his CV, he has an associate arts degree, and his major was music and recording technology from the Madison Media Institute. So I thought that was fantastic. And with that, please come forward. Dr. Johnson's first.
Thank you. Appreciate it. Thank you. Good morning, everybody. It's a pleasure to talk to you today. When Dr. Asana called me about 10 days ago and asked if we could sub for this, I jumped at the opportunity because it's always fun to talk about our research, and especially the findings we're going to show to you today because we think they're pretty exciting, and they're somewhat game-changing in the way we think about pre-symptomatic Alzheimer's disease. And as Dr. Trowbridge was talking about our CVs and accomplishments, I couldn't help but think that all of this that we do and accomplish in our center is possible because of the amazing infrastructure and the amazing people that we work with. It truly is a team effort across the board. Pre-symptomatic Alzheimer's disease is a fairly new concept, and in previous talks that I've done in this forum and elsewhere, we've talked about people at risk or risk-enriched cohorts, and we're now at the point where we can more definitively talk about preclinical or pre-symptomatic Alzheimer's disease, and I'll tell you what that means in just a second. Here's my disclosures, our disclosures. The tau tracer, which we'll tell you about, is provided by Cerveau Technologies. They also fund a study that started recently, and Roche Diagnostics has given us a spinal fluid assay piece of equipment, which is not part of today's talk. Here's some learning objectives. We're going to talk about the biological research framework for Alzheimer's disease. We'll talk about the relationship between biomarkers for AD, AD being Alzheimer's and cognitive decline, and talk about how we can learn more from these biomarkers than we thought we could previously. But before we dig into our research, I just want to orient you to our Alzheimer's program. Just take a minute on this, which is really comprised of these two entities, the Alzheimer's Research Center and the Alzheimer's Institute. It's the same people at both places, but in one of our camps in the center, we're really focused on NIH-funded research. We're providing infrastructure and cores to investigators, whereas in the Institute, we're focused on physician education, community outreach, and raising awareness and the expertise of providers and the public. And both of these institutes really work synonymously with, or very synergistically with each other. In the ADRC, we have a clinical cohort of patients with Alzheimer's dementia and mild cognitive impairment and unimpaired individuals. In the Alzheimer's Institute, we have the Wisconsin Registry for Alzheimer's Prevention, which I'll be talking mostly about today. Presenting these things in columnar format kind of gives the wrong impression that we might be in different lanes. We're really focused on the issue at hand, and the institutional silos are usually more of an encumbrance than a help, but the infrastructure is really tremendous here. It's very integrated, and it's fun to work in this kind of situation. Let me now just talk about the problem of dementia. AD dementia, or Alzheimer's dementia, is the most common form of dementia. It affects about 5.8 million people in the U.S., and about 10% of these are between over age 65, but as you increase in age, like up at age 70, it's about 14% of the population. At age 85, it's about 32% of that surviving population. It has a really tremendous economic impact. It's both underdiagnosed and misdiagnosed in the general community, but more particularly in minority groups. I will say that any estimates like this of AD dementia are somewhat inaccurate because of some of the things I'm going to show you next. It's all of this is based on clinical diagnoses rather than autopsy or research diagnoses. And why that's important is was conveyed by this paper from Ron Peterson up the road at Mayo Clinic, where he points out that amyloid beta, which is this one in green here, and tau, these two proteins are the ones that are involved in Alzheimer's disease. But these aren't the only two proteins that cause neurodegenerative disease. We have something called TDP-43, which is often referred to as hippocampal sclerosis. We have alpha-synuclein-related pathology, which is manifest in Parkinson's disease, neurodegenerative disease, and also something called Lewy body disease. All of these things are more common than you might think, and they all can co-occur with vascular disease, which is probably the most common brain disorder of aging. An individual can have all of these pathologies or something more rare, or they can have a predominance of one or the other. And often in the clinic, it becomes difficult to disambiguate these things. We're going to focus on Alzheimer's disease because that's what we can measure, and that's what we've been focusing on over the last couple of decades. Alzheimer's disease, today you'll hear us talk about A, T, and N. A stands for amyloid, T for tau, also neurofibrillary tangles, and N for neurodegeneration. And the reason we are boiling this down to those three letters is that this is part of the new research framework for Alzheimer's disease that is defining this disease by these biomarkers rather than the clinical syndrome. So this is what amyloid looks like on a neuropath exam, this background here on the left. And then in the inset on the left is an amyloid brain scan of a living individual. And the areas in red are areas where there's amyloid proteins building up in the brain causing these plaques. And on the right, we see indications of neurofibrillary tangles, which are full of tau pathology, the protein tau, and an analogous PET scan where the red and orange areas signify areas of amyloid or tau buildup in the brain. We've become pretty confident in that signal that I just showed you on those PET scans. As a field, we've become confident in that such that there is now a biological definition of Alzheimer's disease based on biomarkers. These PET scans are one credible biomarker. It's also possible to get this information from spinal fluid, and hopefully soon it'll be possible to get this information from plasma so that we can do this kind of research in more of a community and epidemiology setting. Well, with this NIA research framework, we have different diagnostic possibilities. If you're negative for amyloid and tau, then you're normal, of course. If you're positive for amyloid in any form, you're in the Alzheimer's continuum. If you have both amyloid and tau positivity based on thresholds, then that meets the biological definition of Alzheimer's disease. And we would then put some qualifiers on that about whether the person is cognitively unimpaired or asymptomatic, or has mild impairment, or has dementia. So previously, and I still might leak this out today, we talk about, previously we would say Alzheimer's disease, and we meant the dementia stage of Alzheimer's disease. I want to have us all change the way we talk about Alzheimer's disease, such that Alzheimer's is about the biology, whether a person has plaques and tangles in their brain. And then we'll talk about the stage of dementia, MCI, or asymptomatic. Again, here's a couple of examples of some PET scans that we've done locally. The top row are 3 views of someone who has a tau PET scan, and these areas in red are areas, again, where there's tau buildup in the brain, including the hippocampus and the frontal lobe in this person, and their temporal lobe, anterior temporal lobe, and ventral temporal lobe. On the bottom is the 3 views of an amyloid scan from this same individual, and that shows areas where there's a predominance of amyloid, and this can occur in different places. In this particular person, amyloid is more predominant in the frontal lobe and in the temporal parietal lobe. We could have a whole session on how we can use this clinically, and here's an example of that. I'm just going to dwell on this just for this slide. Here's 3 cases that clinically we diagnosed with Alzheimer's dementia based on the best available clinical information, but without these fancy PET scans that we're able to do now. And in 2 of these cases, we were wrong about their etiology. They have dementia, that's easy to spot, but the biology causing that dementia wasn't Alzheimer's in 2 of these 3 cases that finally did have a PET scan. In this 3rd case, you can see loud and clear that there's amyloid in this person's brain. And this, in the best academic centers, we're wrong somewhere between 17 and 25% of the time, and the probability of being wrong increases as a person ages and as other comorbidities have a chance to emerge. And it's not necessarily that we're always wrong, but there may be something else that is the predominant cause of the dementia or the cognitive problem. Well, we're going to leave that and focus now on preclinical AD, and to study those kinds of biomarker changes before the diagnosis. We argue that you have to start early. If the average age of the dementia diagnosis is in the mid to late 70s, and if the lore that we hear in the media is true, and if this disease starts 20 years ahead of that, then we need to start in a person's mid-50s. And that's exactly where we are with the RAP study now. We've started watching people in their mid-50s, and the mean age now is about 67, and I'll focus on that here. But this is the concept that aging is here on the black line, abnormal aging is on the red line, and while a person remains above this clinical threshold, the Alzheimer's pathology of plaques and tangles is accumulating. And when it reaches a critical level, a person gradually progresses from pre-symptomatic to symptomatic Alzheimer's disease. And in order to prevent this disease effectively, we need to start at this early stage to halt the pathology from accruing and to preserve cognitive function. So the Wisconsin Registry for Alzheimer's Prevention is a wonderful study. I had the privilege of working with this since I came to Wisconsin in 2002. It was run by Mark Sager, a geriatrician here for years, and I inherited the study in 2014 when he retired. Here's some features of RAP. 67 years of age right now, about 70% women, about 73% have a parent with Alzheimer's disease, and 40% would be categorized as obese, 40% with high blood pressure, 10% diabetic, 50% with high cholesterol, which is, all of these stats are fairly common for our state for this phase of life. 41% have the ApoE4 allele, meaning those people have the risk gene for Alzheimer's disease. And that's significant because in the general population, it's about 20%. And we've doubled that in this cohort, so it's risk enriched. Here's, on the right here on that map are the individual locations where our participants live. There's 3 data collection sites in La Crosse, in Madison, and in Milwaukee. And here's some other demographic features of the study. There's about 1,580 that have signed up for the study. About 1,100 remain active in it. Our major goals are to identify AD preclinically, and we'll show you some results with that, but also to identify the biological factors like a person's genes, their health factors, and lifestyle factors that might boost their resilience or increase their risk for developing Alzheimer's. And we've heard about all of these things in the media. Every other day it seems like there's something about, does sleep prevent Alzheimer's disease, or a Mediterranean diet, or physical activity, or controlling your stress level, or your neighborhood. All of these things have been associated with Alzheimer's disease, and many of these things we've done locally here in our cohorts. But whether these things impact the Alzheimer biology itself is something that really needs to be studied in detail, and we'll show some results with that. So in order to answer the question, does your favorite item on this list prevent or delay AD pathology or its symptoms, we really need to utilize a study like RAP and our ADRC and look early in life. And so that's where we're going to go now. I'm going to turn this over to Dr. Betthauser, who together with Rebecca Kosick, a senior statistician with our group, have really done some remarkable work. And this is complemented by some new work that Carly Cody in our lab has done. She's a graduate student with us who's here today. So we'll be trying to address some of these questions about the midlife presence of Alzheimer's disease as deduced by these biomarkers, and what can we do with these biomarkers more than just saying yes or no, it's present, and how do they relate to tau pathology and eventual cognitive decline. So with that, let me turn it over to Dr. Betthauser to show us some data. Yep.
Okay, can everybody hear me all right? Get some thumbs up. All right, so I'm just going to present the results from a couple of the studies that Sterling mentioned, one of which I led and the other that Dr. Kosick and I worked on together. And the first is looking at Alzheimer's imaging biomarkers in the sense of whether or not you have it or not. So we refer to this as like dichotomous variables or positivity, negativity. And so it's kind of what can we learn about cognitive abilities and other factors relating to just kind of a status of yes or no. So to get this status, these are exemplar PIB images, which measures amyloid plaques. So on the left, we see a negative case, and on the right, we see a positive case. And we can essentially say yes or no based on a visual look at the data. But we can also use that visual rating to inform at what level in our quantitative measurement of amyloid are people likely to be positive. And so we can see on this plot on the right, so this is showing people's amyloid accumulation over time. And what we can see is that, so for those above this dashed line, which is our threshold, people that are above that line generally continue to accumulate amyloid. So it's we'll get to this idea of chronicity later, but the idea is that once you start accumulating amyloid, you really keep accumulating amyloid without any kind of intervention. And then conversely, for those that are below the threshold, they tend to stay negative, so they don't accumulate amyloid over a period of time. We can do the same thing with our tau PET imaging. So this is an MK6240, or 3 cases, 3 MK6240 scans. And we focus on what's called the entorhinal cortex, and this is a part of the brain that's really involved in memory especially. And it's also important because we know from neuropathology studies that this region is thought to be one of the first areas in the brain to accumulate tau tangles. And so we can do some comparisons with the amyloid negative group and come up with a threshold for tau positivity, again, based on our quantitative measures of tau level based off the PET scans. And we don't have much longitudinal data yet to show whether or not people stay tau positive or not, but that's coming in and so far is pretty convincing. So if we kind of look at across RAP and ADRC, at just people who have had PET scans and figure out whether they're amyloid positive, tau positive, and the 4 combinations of those positive and negative categories, we can see that by and large, most of the people, about 74%, are actually negative for both amyloid and tau. Whereas people that are tau positive only is about 3%. People amyloid positive only is about 13%, and only about 10% of the participants are positive for both amyloid and tau. And to contextualize this a little bit, Sterling mentioned we're really trying to focus on this pre-symptomatic phase of Alzheimer's disease. 92% of these people are cognitively unimpaired, so they're not showing any clinical signs of impairment in terms of cognition or function. Whereas, about 5.5% have mild impairment and about 3% have dementia. So the first study I'm going to present is from RAP. So these are all participants that were cognitively unimpaired when they began the study. And we had about 8 years of data before we had available PET imaging. And the thing that we were really focused on looking at was, in this 8-year period before PET imaging, did people, in terms of their cognitive performance on some of these tests that we give, did they differ based on their biomarker status? So did people with both amyloid and tau decline any faster than people that didn't have amyloid and tau, for example? So to do this, we dichotomized people into those 4 groups that I mentioned previously. We used a composite that is a measure of memory and executive function, and it's thought to be a bit more sensitive to early changes in cognitive ability. And then we used a statistical approach that would allow us to determine whether or not people were declining faster over time given their particular biomarker group. And then we also looked at some demographic comparisons of age and other factors that we know are associated with risk of dementia, and including also some health factors. So in terms of the demographics, participants at their most recent cognitive assessment were around 65 to 70 years old on average. And so if you remember, we had about 8 years of follow-up, so that means the study was really started when people were kind of in their late 50s. And if we look across all these biomarker groups, we really see that there really are not that many demographic differences. So if we look at sex or race or family history of dementia or a measure of verbal intellectual attainment, none of these things were different between any of these biomarker groups. One of the things that was different, as Sterling mentioned, was this ApoE4 gene. So this is a risk gene for Alzheimer's dementia. So we see that people that are amyloid positive and tau positive have a higher frequency of having this E4 gene. But importantly, this is, again, it's a risk gene. It's not deterministic so far. And we can see that here, which is in the people that have this E4 gene, or E4 allele, sorry, 60% of these people don't have amyloid in their brain based on PET imaging. So again, it just kind of indicates that E4 is a risk gene for developing amyloid and Alzheimer's dementia, but it's not deterministic so far. If we just look at some average images of the different biomarker groups, so this is the group that is negative for both amyloid and tau, tau only, amyloid only, and amyloid and tau positive. A couple of things pop out. So I mentioned the entorhinal cortex being that region that we think is earliest to develop tau pathology. And we can see in this tau-only group that really that's the only area where we're seeing elevated signal with our tau imaging. And as we look to the double positive group, we can see that that tau is now spreading throughout the rest of the brain. And it's not just this middle temporal region that's being implicated, but really it's spreading throughout other regions of the brain. We can also see that in these double positive groups that they also have higher amyloid than any of the other groups that are positive for either just one biomarker or none. So if we look at people's cognitive performance over time, so these are commonly referred to as spaghetti plots, and what it's showing you is effectively people's cognitive score on the y-axis, and on the x-axis is their age. And what we're really interested in is the slope of those lines, so how fast they go up or down over the course of time. And the big thing that jumped out to us in the data was this double positive group, that these lines are really going down rather quickly compared to these other groups. And another thing is that these lines are going down well before we're seeing what we would call clinical impairment. So this is that, again, that idea of pre-symptomatic or preclinical Alzheimer's disease, where there are changes happening that are likely due to disease, but these aren't reaching clinical levels of impairment. With some of the more formal statistics, we can see, again, that there were group differences between these biomarker groups, and that those that had both amyloid and tau were declining much faster than those that had one or no biomarkers. And if we look relative to our control group, which would be the amyloid negative, tau negative group, the group that had both positive amyloid and tau were declining about 3 times faster in that period leading up to PET imaging. And I think this is a really big point that we're starting to understand is if we have positive biomarkers, what does that mean for somebody's cognitive ability? And this idea that decline is going much faster if you have these positive biomarkers. If we look at a couple other factors, so neurodegeneration, for example, we can look at in the hippocampus and look at the volume of the hippocampus or the size. And we can see that there's a small difference between those that have both positive amyloid and tau, but there's a lot of overlap between this group and the other groups as well. So it's a little harder to say whether or not they're neurodegeneration positive, for example. And another important finding was that if we compare health and vascular features either at the beginning or at the end of the study period, we're not seeing really any meaningful differences in terms of things that you might associate with risk for dementia. So BMI, waist-to-hip ratio, measures of cholesterol, blood pressure, those kinds of things. So none of these really differed between these biomarker groups. And I'll have some of Carly's results to drive that home a little bit more. So the summary of this study, again, this was persons that are relatively young in terms of aging research. So again, starting about the age of 59 or 60. We're seeing that amyloid and tau positive biomarkers are strongly associated with cognitive decline. They tend to have a higher proportion of E4 carriers. And really, I think the important thing to drive home for clinical utility is that these are likely the people that are going to come to clinic. This is the group that is probably at the highest risk of developing cognitive symptoms during their lifetime. And I think the other important finding from this is, compared to other studies that have come up with these hypothetical models for staging of Alzheimer's, this study really suggests that these cognitive changes are happening much earlier than maybe we previously thought in the community. And lastly, and I'll have a little more data on this at the end, but we're so far not seeing any evidence that vascular factors are contributing to the pathology in any meaningful way. And so the second study is taking a little bit different look at this. So the first study, we were looking at yes or no. Do you have amyloid? Do you have tau? And that's a bit of an oversimplification. So we know that, for example, if we were to look at somebody's cholesterol, right, there's ranges of risk. So if I have cholesterol in the normal range, I have low risk for cardiovascular problems, right? But as that level goes up and up and up, I start to see more risk of cardiovascular events. So we kind of took that approach with our Alzheimer's biomarkers with amyloid. And again, to kind of point out in these longitudinal plots of the data, this is really a chronic process. And people that start accumulating amyloid, they don't go the other way. They continue to accumulate amyloid. And I'll show some data in a minute to show that kind of some of the effects of that continued process. But there is a lot of difference in when people start to accumulate amyloid. So we can see on this plot, for example, we have a person here in their early 50s crossing over this positivity threshold, and again, somebody over here that's age of around 78. And this is almost a 30-year time window in somebody's life that they may or may not start to develop amyloid. And in order to understand that and understand the risk that it means for developing cognitive symptoms in life, we need a way to kind of quantify the chronicness of disease or the duration that they've been exposed to amyloid. And so that was really the motivation for this second study that I'm going to present that Dr. Kosick and I recently are in the process of publishing. And so we can use a method called group-based trajectory modeling. And really all this is doing is we're taking those amyloid by age patterns, and we're looking for groups. So groups of individuals that seem to be kind of on the same path with respect to age. And so if we do that, we can pick out these 3 accumulating groups. So they're shown here in the blue, green, and red. And we can actually use some statistical models to show how those people develop amyloid over time with respect to age. And importantly, we can use that information to estimate when each of these groups will become amyloid positive or did become amyloid positive. And we can apply some probabilities, and we can actually estimate the age then. By applying that to individuals, we can get at within a person to say, when do we estimate that they became amyloid positive? And so effectively what this allows us to do, so here we see the kind of those different group functions, the blue, the green, and the red. And we can remap that time axis onto how long we think they've been amyloid positive. And when we do that, we get a much more homogeneous curve, meaning that as we progress and increase in levels of amyloid, most of these people are really seemingly accumulating amyloid at the same rate, or they follow the same pattern with respect to what we think is disease time. And so what that really allows us to do is to take, for example, a single scan where somebody, say, comes in with this quantitative measure of 1.5 amyloid. And we can actually map that onto a time of how long we think they've had amyloid. So in this case, slightly over 10 years, for example. And by being able to do that, and by being able to estimate when somebody became amyloid positive, we can start asking some questions in a little bit different way, and some really important ones for understanding people's risk of developing clinical symptoms. So for example, we can look at things that would maybe influence the age of onset of amyloid, or the relationship between the duration that somebody has had amyloid and their cognitive performance, or we could look at relationships with tau or other biomarkers. And we can really start to probe some of these health and lifestyle factors that might affect the pathology, but might also affect cognition. And we don't have much on this resilience topic quite yet, other than I'll have a case at the end. But it does allow us to start getting at what might help people to develop either AD pathology or cognitive symptoms for a given level of amyloid. So this first study, again, was from the Wisconsin Registry for Alzheimer's Prevention. And effectively what we looked at was, using this chronicity approach, we can say, how long did somebody have amyloid when they started in the study? And then we can look at how they performed on their cognitive tests over time. And when we do that, we can see a really strong relationship between the duration of amyloid that they had when they started in the study and how they performed over the next roughly 8 years. So we can see this group that doesn't accumulate amyloid here is this black line. So they're performing a little bit worse over time, but it's a really slow decrease over time. Versus somebody in this blue line would have amyloid for, say, about 10 or 15 years at the beginning of the study. They're declining much faster compared to these other groups. And we see that these patterns start to diverge or become distinct from each other somewhere in the age range of 70 to 75, depending on how long you'd had amyloid at the beginning of the study. If we look at just kind of some clinical endpoints and some research-based endpoints, we can see that this measure of amyloid chronicity, or the duration of amyloid, gives us a better risk, or better explains the risk of progressing to MCI or AD, or crossing some norms that we've established in the RAP study for abnormal cognitive performance. And then another slide that is really relevant for clinical diagnoses is if we think about the level of amyloid as relating to a moment in time, and then we look at clinical status, we can see that, or we're starting to see some evidence in our most recent data that there is kind of there's likely a critical point at which if you have a certain amount of amyloid and you've had it long enough, that you're likely to be impaired. And this is something that we're actively pursuing to understand more of that, how much risk there is of having clinical impairment. We can shift gears a little bit and look at tau pathology. So if we, again, focus on that entorhinal cortex region, that memory center, we can see that we really don't do a good job of trying to figure out who's going to have tau based on just somebody's age. But if we realign that time axis again in how long we estimate they've had amyloid, we can see that this pattern becomes a lot more clear, and based on our current data, it looks at about 10 years into developing amyloid, people start to also develop tau. Another thing to point out is that as we increase in levels of amyloid, it's not just the level of tau that we're increasing, but it's also that spatial spread. And so that's effectively what this plot is showing, is that for low levels of amyloid, we're really only seeing small increases in these early regions associated with tau deposition. But as we get more and more amyloid and we move higher up in these lines of amyloid, we're starting to see other brain regions affected by tau as well. And this is, I think, another really interesting finding that we're sorting out recently, which is, I mentioned, E4 being a risk factor for developing dementia and Alzheimer's disease. So one of the things that has been really interesting in our data so far, and we're starting to see being replicated in other centers, is that the rate at which somebody accumulates amyloid is not really different between those that have this E4 allele and those that don't. And more recently, we took a little bit of look at this to see how these groups differ in terms of when they start to accumulate amyloid. And so, for example, if we have people with 2 E4 alleles, we estimate that they're becoming amyloid positive in their mid-50s, whereas people with 2 E3 alleles, which are not associated with AD risk, are becoming amyloid positive in their early to mid-60s. So this is about an 8-year time difference in somebody's lifespan, and we might suspect if we have a 20-year process to develop amyloid, that the group that's E3 carriers might die of some other cause before they actually show clinical impairment, whereas the group that gets amyloid younger would have more risk of developing cognitive symptoms during their life. So to summarize this part of the study, this heterogeneity in amyloid positivity is really largely explained by the age of onset of when somebody develops amyloid. We're seeing that these accumulation patterns of amyloid are really quite consistent. Once you get on the amyloid accumulation path, that path is really consistent between different people. We're seeing relationships with amyloid chronicity and cognitive decline, and with tau in ways that are intuitive. And the E4 finding, I think, is something that will be really helpful and will also inform how we can study some other factors. For example, we might suspect that somebody with some kind of socioeconomic disadvantage might have exposure to certain environmental factors, and that could also maybe influence this age of amyloid onset. But these are things that we need to look at further. So for the last study, this is work that was performed by Carly Cody. So she's a graduate student in Sterling's lab. And the goal of this work was really to take a little bit closer look at the relationship between some of these health and what we call modifiable factors, so things we could change, and cognition, and then also this amyloid duration. And so to do this, Carly used what's called the Libra Index, which is the lifestyle for brain health. And this was derived as a measure of the potential for somebody to be resilient, essentially, to dementia based on a large analysis of many studies. And so we have various factors that go into this score. They include things like alcohol consumption, cardiovascular disease, smoking, obesity, hypertension. And what Carly did was looked at whether or not this risk score, so these health factors, whether or not those were associated with risk of incident cognitive impairment. And what I mean by that is whether or not people that had, say, higher Libra risk were more likely to hit this threshold for cognitive impairment compared to those that have, say, low risk. So we do see that pattern in the data, such that people that have a low risk for Libra are hitting this threshold for incident impairment later on compared to those at high risk. But that's also not really the full story. So of these, this was a study of 1,244 rat participants. 236 of those had amyloid PET scans. So we applied this amyloid chronicity concept to the same data. And what effectively we saw is that if you had a higher risk with Libra, you had lower cognitive performance overall, but it didn't affect the rate at which you were progressing over time. Whereas if you had high amyloid at the beginning of study, you were declining faster compared to somebody who didn't have high amyloid. And there was really no relationship between Libra and this duration of amyloid. And so if we think about that kind of in the context of the survival analysis, so the blue lines are people that are at low risk, low Libra risk, and the red are high Libra risk. And then the dashed lines are people that are effectively amyloid negative when they start the study. The solid lines are people that are amyloid positive for about 10 years. And what we can see is that if we compare, for example, the red and blue lines within a given level of amyloid, those lines go parallel to each other. So it's not the case that somebody that has high Libra risk is performing worse faster than somebody that is at low risk. But if we instead contrast the amyloid, so if we look at this blue line versus this blue line, for example, we can see that people who have had amyloid longer are declining much faster than people that are amyloid negative. So, so to conclude, overall, we're seeing pretty strong evidence that having both amyloid and tau is associated with deleterious cognitive deficits. This concept of amyloid chronicity is really something that's new and a useful tool that we can really start to probe the data in a little bit different way and start to understand some of these future risk ideas and start to more directly investigate things like resilience. And just to kind of leave you with a little bit of a thought on this too, many clinical trials are starting to incorporate amyloid positivity as an enrollment criteria when they're doing these treatment trials and things. But I hope maybe that our data has convinced you a little bit that this might not be sufficient, because amyloid positivity could mean you've been positive for a year or 20 years, and you know, your prognosis is going to be different probably if you're just starting versus you're towards the end of that cycle. So for future directions, we plan on looking at this with other biomarkers and really trying to understand risk in the background of all the other things going on in the brain. So we can look at in kind of a bubble at, you know, just the relationship between amyloid and your risk of clinical impairment, but we know there are other things happening in the brain. And if we start to incorporate those, we might get a better sense of somebody's risk of developing impairment during life. Another real important concept is this idea of resilience, or your ability to kind of thwart the disease by some factors that maybe we can alter in life. And there is some interesting stuff here. I mentioned the E4 business showing that it's changing when somebody develops amyloid, but we also know there's a lot of people that don't develop amyloid. So we have to start to figure out why people develop amyloid at certain times and why people don't at all. And then moving on, we really want to start translating this to fluid and so cerebral spinal fluid and blood plasma biomarkers. And this will really help, I think, clinically, if we can do this in a similar way. We can have a test that's one, more affordable and far more accessible for a whole bunch of people. And this could really be helpful clinically, I think. And then so to summarize, or to kind of wrap this up, so I've painted this picture that seems deterministic, right? That if you have amyloid and tau, you're going to have cognitive deficits. But on the concept of resilience, this is a case that came in recently. So this is a 63-year-old woman. She came into the RAP study for PET imaging at the age of 64. We can see her amyloid scan is floridly positive. Her tau scan is floridly positive. But she's incredibly high functioning. Her baseline IQ was 124. And she's had 10 years of RAP visits looking at her cognitive abilities over that previous period of time. And she's rock solid over time. She has not deviated. And this is really an interesting case because based on the numbers and everything that we think, she's estimated to have amyloid for 18 years. And if we look at other people in that same range, most of them are impaired, but she's really not showing signs of impairment. So these are the kinds of things that we need to better understand if we're going to have some hope of having people be a little bit resilient to the cognitive changes due to Alzheimer's disease. And on that note, I'll wrap it up. I think in addition to Sterling's comments about the support we have here in Wisconsin, I think it's really important to acknowledge all of the participants. I've done site visits. I did a lot of the PET imaging stuff early on. This is a big commitment on people's time, not only the people contributing to the studies, but in terms of staff and faculty, but also the participants that make this work possible. And we're really hopeful that this will really help us understand Alzheimer's disease moving forward and something we can translate into the clinic. So, thank you.
Okay, with that, I will open it up to questions. We'll have both people come up front. Dr. Bussey.
This is a great example of the power of imaging to give us an idea of the biology of the disease. Toby, if I could ask you a question, you sort of alluded to it. What determines sort of when either the plaques or the tangles get deposited? Is there so many people are at risk for their life, but what seems to determine when these things get deposited in the brain?
I don't think we know.
Yeah, so the question was effectively what things determine if amyloid is going to aggregate and when. And I think we're really in the infancy of understanding that. I think we're starting to see, this E4 risk effect is definitely a contributor. That gene is tied to a bunch of processes in terms of creating amyloid in the brain, for example. But I think there's a ton of work we have to do to know exactly that question, right? Of why is it that somebody starts developing amyloid in their 50s versus their 70s, and what's going on in the brain to drive that process?
If I could add to that, this work is establishing that endophenotype. Now we can look upstream from that more effectively and hopefully see what's causing all this.
Dr. Schultz.
Do you have any data to indicate that there's a difference between what IQ you start with at a certain age and what the trajectory of the decline would be?
That's a great question. It's about your starting point cognitively, your baseline IQ and your cognitive resources, and if that influences the trajectory of this disease. It definitely does influence your cognitive trajectory and your clinical trajectory. The higher functioning people maintain function for longer. They have more resilience or more buffer, but their amyloid pathology is accruing at the same pace. But they'll manifest their symptoms a little bit later because they have this extra buffer.
In that.
I was just curious, are there a group of patients who you would clinically describe as having Alzheimer's disease, don't have amyloid or tau, and no other obvious explanation for their dementia?
They would be.
Repeat that question.
Okay, the question is are there groups of people or a patient that would be considered to have Alzheimer's disease but not have biomarker evidence of amyloid or tau? Is that the question?
Yeah, and sort of the follow-up to that is whether you've kind of created a tautology that if you don't have amyloid and tau, you don't have Alzheimer's disease, that you're sort of defining it that way, and they're really biological correlates, and in terms of what's going on in a causative way, it could be a factor not yet described.
That's a great point. The question is, the add-on question is, are we creating a tautology or a circular arrangement here where we're defining this disease based on these two proteinopathies, and is that limiting our discovery? This is a research framework, but it's based on the neural path diagnosis. When you come to autopsy, a person comes to autopsy, the pathologist diagnoses Alzheimer's based on the presence of plaques, amyloid plaques, and neurofibrillary tangles. So that's the approach we're taking in the research setting in this biological definition. So by that definition, a person doesn't have Alzheimer's disease if they don't have biomarker evidence or eventual neuropath evidence of the disease. And that goes back to that first slide, one of those first slides I showed with that radial figure with other proteinopathies. We know that there certainly are other things, and they look just like Alzheimer's. TDP-43 pathology presenting at the clinic looks just like clinical Alzheimer's disease, and Lewy body disease often mimics Alzheimer's disease clinically, but the underlying pathology is different, and that's what this framework is trying to do, is improve our construct validity of what we're doing. But that is a potential danger. Once you commit to something, you cut off some of those possibilities of what else might be explaining it.
Thank you very much. Any further questions?